And I think we're now recording. So this is week 12. Can you believe? Week 12, and this week we're doing the exam tree and lab practical tree. There's a couple of other things that I do want to mention. I really appreciate you both making it here early, like first thing of the week. And I also want to go through the material, so if you have other appointments, other classes, you don't miss anything. But I really do appreciate you making it here. And I do make a note. Trust me, when I go through the final grades, I look who's been around and who has made everything they can possibly do to make sure that they're gonna do well. Just let me open a little bit of a door. Keep going. Door for my cat. She's stuck from one room to another because I'm trying to keep the house warm by having doors closed. Like I said, we're totally snowed in. I live on Mount Lemmon foothills, so we got a pretty good snowfall last night. If you're somewhere where it's a bad weather, be careful with driving. It's not you guys that I worry, it's the other drivers, and then that causes problems for you guys. The snow can be a surprise for some. But what I wanted to do, just jump in to what we're doing today. I wanted to start by kind of an announcement. I think this is something familiar that you might remember from our earlier discussions, but I just want to make sure that everyone has all the information. So for the last week, which was week 11, notice that you still have time to do those assignments until end of this week, I think. And same goes for the week before that. And that's because there was all sorts of students have a lot of things going on now. And I think that there was a little delays with publishing something and so on. So I totally get it that I just want to give you time that you can get all the possible points that you can get. And notice also for this week's exam and lab practical, it's actually 2 weeks that I've allocated for it. So if you need longer than this week, you have also next week to do it. Of course, I always encourage you to try to stay on top the best you can on the workload. But I really want you to have a chance, for example, before taking this exam or taking this lab practical, to have done the homework for that or have done the assignments for that. So you're not doing something that you haven't had a chance to practice yet. So just notice that there's more time, and same goes for the any other assignments. If you have later on or with these exams, with these practicals that you need extra time, just let me know and we'll figure it out. I'm always trying to be as flexible as I possibly can with the due dates, because things happen. I know how busy life is, and I really think this shouldn't be a test of time management, although it is one of the skills that we learn. But I want you to have the knowledge that I'm not out to catch you with just making something unachievable or uncapable in terms of the timeline. So there should be plenty of time. If you need any more time, you just let me know. But that's something that I really wanted to highlight, that you have for last week and the week before, still end of this week to do the assignments, and then for this exam, this lab practical, this week and next week to do those. In terms of what we're doing today, we're really focusing on the exam tree review study guide. And if you have a chance, I encourage you to either download it from Canvas or print it, or if that's not your way of learning, then you can just listen, or if it's your way of learning that you take notes, whatever works for you. I heard, I appreciate it. I already heard from one of our students here today, this morning, that you were able to access the study guide. I'm just putting the final touches for the exam, so it will be up and open probably in about 2 hours. I just want to make sure that everything is actually covered on that study guide. So no curveball questions. And that's really where we're focusing today. And I want to leave time also if you have any questions, anything that I can help with. I know that there's been a lot of new ways again doing assignments for last week's assignments. It seems that we always have something new to throw in. But what I really wanted to highlight, I'm probably more excited than you guys are. I hope that that's not the case. I hope that you are also excited about the fact that we're approaching end of the semester. I can't believe where it's gone. It seems like ever since the COVID changes happened, things are just happening rapidly. So this semester is ticking along nicely. And after this week, all you have left is 3 chapters. That's all we have. We're going to talk about digestive system. We're going to talk about reproductive system for males and then for females. And that's it. And for the week 16, we'll have an exam that covers those 3 chapters, and then we have our finals exam. And like I promised you, finals exam is something else than just you memorizing stuff or having to study as hard. It's more about you being able to produce whatever you think is the best way for you to demonstrate your knowledge of the material. And I have the instructions of that. I think we'll look at them, but we'll get through this week first. But what I really want to say, you're amazingly far already. You're here and you're hanging on, and that's really the biggest trick on these online courses, to just not give up. It's really easy. There's so much going on. You just at some point let things slide, and then catching up becomes really difficult. Like before, with the previous exam weeks, previous lab practical weeks, there's no actual assignments other than the exam and lab practical. And also remember that you can do the assignments from the past if you need to catch up on something. Like I said, I'm opening all of the modules as much as I can. So that if you have one of those codes on Connect that's about to expire, you can try to get them done. But also I do have, and again, that's another thing that I'm just putting finishing touches. I do have alternative assignments if your Connect code runs out. But I really do recommend that you try to use that code if you can, because the alternative assignments, they're a little different and they might be a little bit more laborsome. Believe it or not, than doing the Connect assignments. This week, like I said, the exam and lab practical 3, and they are open for 2 weeks. So this week and next week, notice that the exam is 100 points, lab practical 50 points. They add up quite nicely when you get those done in terms of your final score. I think that that's all similar to what we've done, with the exception, like I said, that this week you've got twice the time, or you've got this week and next week to do all the assignments. There should be the study guide for this week's material available. And what I'm going to do, I'm going to pull up that, and we can have a look of that together and discuss if there's anything that you think that might be kind of unclear, or then alternatively, we'll just go through what are the key points that we really want to know about this chapter. Let me just jump to the right window. Or these chapters, not just this chapter. So here we are. You should be able to, I hope, see the study guide for the exam 3. Oh, I didn't mean to go there. I just tried to make it as big as possible. And if you do have the study guide printed, you have it downloaded, that's great. You'll be able to follow along. If at any point I go too fast, you just let me know, and I'll either slow down or I'll go back. I don't want you to feel like you're having to race. This is not a speed test, or preparation is not about who's the quickest. I do also want to mention, I'm sure you two know, but all of this recording, or all of these sessions I'm trying to record, and they will be available then on the Canvas with just a short delay. So we're going to start with chapter 22, which looked at the respiratory system. And this first part is really just kind of getting us oriented, how things are linked in the body, how different systems work together. So I really think that the big message from here is just that, hey, one system cannot exist alone. There's a link always to another system. And if we just kind of quickly look at what we have already talked, we looked at the cardiovascular system. Well, cardiovascular system, in terms of if we think of how does it relate to the respiratory system, well, it's the transportation system. So it transports the gases, it transports the nutrients, waste products, hormones, all sorts of things. So without cardiovascular system, the respiratory system would be kind of redundant because yes, we would get oxygen to the alveoli of the lungs, and yes, we might have carbon dioxide, but we would not be able to take the oxygen anywhere from the lungs to where it's needed in the tissues, and we would not be able to take the carbon dioxide from the tissues out of the body. So really, you cannot have one without another. We had a look of the lymphatic immune system. They were all about body defenses. And again, if you think you might breathe in stuff that you really don't want in your body, so their function is to protect you. And we looked at the end of, not last week, but the week before, we looked at the urinary system, which again, we noticed that one of the big functions of that was to regulate blood pressure, but it did a bunch of other things. It was about cleaning blood, eliminating materials, regulating the fluid balance. There's going to be a bunch, and we'll have a look of that a little later on this study guide. But the big thing with urinary system was how it relates to the respiratory system is that as a process, we ended up with excess hydrogen ions in cellular respiration. But respiration alone, we might not be able to remove all of the hydrogen ions. So urinary system steps in, and urinary system then eliminates that. That was a really kind of an abstract way just to try to get us thinking that systems are linked. So that probably gives you a clue that that's not probably the biggest question that's going to come up. But really, if we start thinking of the respiratory system, we ended up noticing that there were three things that we're always primarily worried if we get a patient. We're worried about their airway. Is the airway open? We're worried about, are they breathing? And if they're not breathing, can we assist them breathing with giving additional air blowing into their lungs by doing mouth-to-mouth resuscitation? And then, of course, we have the chest compressions to keep the cardiovascular system going. And the order of those changes a little bit depending on who you ask and when these guidelines have been published. But we notice that breathing and airway take two of these three vital things that we want to do. So they're really, really important. And like I said, when we do cellular respiration, we end up, I don't know if this is going to work. Let's see. That was interesting. I thought that I might be able to do it. And when we do cellular respiration, we end up with a need for oxygen, and we end up with a need to get rid of carbon dioxide. So really, the role where the respiratory system, if you think of the big picture of the body, is that it steps in to supply us oxygen and remove carbon dioxide. Because really, if you look at what your body does, what's the main function, what's all your body does, it really is nothing else but cellular respiration. That's what we want to do. And that's all we need to do to keep all the other body processes going. But as a result of cellular respiration, we are left with that excess carbon dioxide that we need to get rid of. And we are in a need of oxygen that we must have a supply into the tissues. So that's the main point of respiratory system. In addition, there's other things. Respiratory system helps with olfaction, which was our sense of smell, also with speech production. So all of these other non-respiratory functions are important as well. And I think olfaction is interesting because it kind of overlaps also with the digestive system, which is the next one that we'll have a look. So again, all of these kind of get connected. But the big message here, if you want to think what could be good exam questions, it would be that the supply of oxygen and removal of carbon dioxide is the big story, the big goal of respiratory system. We have various parts in the respiratory system, and we'll go through those during this review session. But I just kind of wanted to get us, I think it's always nice to have a roadmap or some sort of an idea what's to come. And I wanted to get us thinking of that. So that's why I've listed them all there. Of course, the most anterior part of the respiratory system, and really the first part where air comes in, is our nose, and especially nostrils in the nose and so on. We'll talk about nose a little bit more. We won't go into great detail, but a little bit. And then behind the nose, we have our nasal cavity. The nasal cavity is not there just to be an empty space. It actually does a heck of a lot already before any other part for our respiratory system, or us being successful with breathing and so on. We mentioned the paranasal sinuses. Sinus was always, when we talked about in anatomy, some sort of an empty space, empty gap within an organ, within a structure. If it's paranasal, that refers to this region that we're studying on this chapter. So we'll talk a little bit about paranasal sinuses, mainly because it's something that is still not fully understood. Of course, pharynx and larynx are structures that I think you're already familiar with, so it's more of a review, but we really do remember that larynx became super important when we think about the voice production, which was one of these functions of the respiratory system, as we saw, that that are in addition to the gas exchange. So we'll talk about that a little bit. Then we have, of course, our windpipe, our trachea. We'll have a look at that. There's a little bit of structural and anatomical adaptations. And trachea then divided at the carina to two primary bronchi. And we'll talk about those bronchi a little bit. We'll go to smaller bronchioles, eventually ending up having a lungs at large, but also at the very end, the alveoli, which was really the site where everything that other parts of the respiratory system have been working for, where that happens. So it's the business end of the respiratory system. So let's have a look at the nose. That's the first one that we will review. And in terms of the nose, really, the big thing is that normally we breathe through our nose. Only occasionally we recruit mouth, but most of the breathing in a normal situation would happen through nose. And that's because of all the things that our nostrils, but also especially the nasal cavity does. Sometimes I get students, especially when it's face-to-face classes, not so much on online classes, which I don't know if it's a blessing or not, but I don't see students yawning. But when they yawn, this might be me just trying to feel good about my life. I never think that, oh my God, they hate me, they're bored, because in my mind, yawning is body's response to decreased oxygen levels, and it's a body's way of getting quickly a lot of oxygen into your body. So I don't know if you've ever noticed when you're in a room where one person yawns and then everyone else starts yawning as well. And we often talk about how yawning spreads. Well, that's really based on what I'm proposing, that because the oxygen levels are so low, that our brain is slowly starting to notify us that, hey, we probably should do something about that. We need more oxygen in the body. And what's the better way to get more oxygen? Because our nostrils are always going to be the same size. Then open up your mouth as big as possible and take a deep breath in. So that's in my mind why students yawn in a class. It's all about just air conditioning. And maybe it's a time for a break. Yawning might have some other psychological connotations and reasons, but not for our purposes. So if I ask that in an exam, that's what I probably would be looking as an answer. We'd already mentioned the nostril, sometimes also nares or nares. You might hear terms being used. Nostril is really the first part of everything. And what you'll notice that nostrils have hair. Some people have more pronounced, some less pronounced. Usually older people, especially older men, it is fairly coarse, fairly pronounced. Some people trim their hair from their nostrils, whatever they do, it's up to these people. But remember that that hair serves a purpose, at least to a certain extent. That's just a filter. That's a mechanical filter to filter the big particles out of the air that you probably don't want to take. That's why, for example, if you've been in a dusty room or working in a wood shop, when you get home or at some point you end up having to sneeze, that's getting rid of these big particles that we filtered, might have gotten stuck into your hair at the nostril or further back. We're trying to remove that. So that's kind of a mechanical way of ensuring the quality of the air. Let's have a look at the nasal cavity, the space behind the nose. Well, what we find that nasal cavity is lined by this membrane. It's not skin anymore. It's a different kind of a membrane, and it's called mucous membrane. Much thinner than the skin, much stickier, much wetter, and this all is because it's lined with mucus, and this mucus is produced by goblets. And the function of mucus is really multiple, but the big one that we want to look at while we were talking about the hair of the nose is trapping particles. But the mucus does also other things than trap particles. It contributes to the humidity within the nasal cavity, the thin wall of the mucous membrane. Well, that means that there's a lot of blood vessels close by its surface. So it's really warm space as well. These blood vessels bring a lot of heat with them. That's why nosebleeds are so easy to get, because this is such a thin surface and there's a lot of blood supply there. But for our purposes and our story of what we want to notice about the nasal cavity, we remember that this mucous membrane served one of the important functions to trap particles. If it's this sticky, kind of humid surface, everything tends to get stuck into it really nice. And then we start worrying how to get rid of it. And sneezing is one good way. Students, I do summer classes with younger students, not college age students, but grade 2 to grade 6. And they're always asking, why do you produce snort? Why do you have buggers in your nose? Well, that's the transition zone where mucous membrane ends and you turn it and it turns into a skin. That transition zone is where a lot of these particles are transported, but also mucus that then just dries up. So that was kind of going off the topic. But anyways, what else we notice in the nasal cavity? One important set of structures that I do want you to know is that we have conchae. And you might remember conchae as these protrusions from the lateral wall of the nasal cavity. So remember, not medial wall, but from the lateral side of the nasal. Wouldn't that be a great exam question? What wall? So we have 3 of them. We have superior, middle, and inferior conchae. And they have 2 main functions. First of all, having these protrusions that come out of the wall, they give us more surface area for this mucous membrane. But they also, because of their shape, they do something that's important for us. They mix up the air that passes through this structure, generating kind of a turbulence, if you wish, horizontal flipped horizontally tornado kind of a flow of the air. And the function of that is that we want to ensure that as much of the air, hopefully all of the air, gets a chance to be in direct contact with this mucous membrane, so we can really trap all the particles that are in it and do all the other stuff that we want nasal cavity to do. So it really serves a purpose that we have as much of this surface area and we also get the air rolling inside this cavity. A few other things, your nasal cavity is divided in two by nasal septum or nose, anterior part, by nasal septum that separates the left and right. When you get a nose job done, for example, we might want to make sure that the nasal septum is still in a good shape there. Sometimes you get people who use heavy drugs, for example, through nose, cocaine, and so on. Their nasal septum completely deteriorates. Boxers as well. But it turns out that most of us, we don't have very regular nasal septum. It's not very straight. It's kind of kinked or just weirdly shaped for many of us. And as a side note, which is not in your exam, but you do know, I hope, that people who get a nose job, your nose can grow back. So that's something to bear in mind as well. Nose is a tricky one. And more nose jobs you've had done, harder it is for the next time. We might need to take extra either cartilage or extra bone, often from the ribs, and then replace. But that's not in your exam. The inferior, the bottom layer of the nasal cavity, the floor base, is made by our palate. We have the hard palate at the front. If you feel with your tongue, that is hard because there we have the bony structure separating the nasal cavity and oral cavity. And further back you go, that turns into a soft palate, and eventually we have uvula there. And we'll talk about the function of that a little bit more in the digestive system chapter. But really, if we think of what soft palate and the uvula do, they do seal off the nasal cavity so that nothing goes up there when we swallow liquids or solids. We try to make sure that they go where they should be going. Now let's have a look what else we can think of that would be interesting to know. Let's talk a little bit about the paranasal sinuses. So we had 4 types of paranasal sinuses. We had our, and we remember again, the sinus was some sort of a hollow space. We had our frontal sinuses within the frontal bones, so above the eye sockets. Kind of where your eyebrows and going a little bit higher from that, where they would be. We have our maxillary sinuses, again, 2, 1 on each side within the maxillary bone. So one on each. And then we have our sphenoid sinus. That's within the sphenoid bone, deep into the head, further back. So that's kind of hard to illustrate, but think of those diagrams that we had and refer to those if you're kind of wondering. Sphenoid bone, all of these bones are really good clues where they are located. So if you spend your time learning the bones, this is really where it pays off. And then we have ethmoid sinuses, or ethmoid labyrinth, as I prefer to call it. It's up to you which term you prefer. I use the term labyrinth because these are multitude of small openings or small spaces within the ethmoid bone. It's not just one sinus. It's kind of a group of interlinked hollow spaces there. So I think it looks more like a labyrinth, but it's up to you which term you prefer to use. But 4 types of paranasal sinuses that we need to know. And their function is the one that I find kind of interesting to consider because some of this we know and some we still don't know. Some of this is still up for debate. So that's why I kind of wanted to bring it up and for us to talk about that. And again, I think that would make a fun exam question. So why do we have these spaces within this region? Well, if you think of the skull is kind of a big structure in comparison to your neck. So it's mainly your muscles of the neck that do the big job of balancing off your head and supporting it so that it doesn't collapse to side, down, or back. And anything that we can do to make that skull lighter would be probably a smart thing to do. So these sinuses, it's fairly well agreed that these sinuses reduce some of the weight that we have in our skull and for our skull. The other thing that we do agree with, and this has been proven with many sorts of measurements, many sorts of tests, how would our voice differ if we didn't have these sinuses? And there actually would be a difference. So your voice is really just a vibrating stream of air. We have this resonance going on in this air, and these spaces give us voice resonance spaces. So they play a role in our voice production for sure. The next one is very much kind of being debated. I can't say for sure if you should believe it or you shouldn't, but we're going to learn it either way. So you might remember that it was proposed that paranasal sinuses might serve a purpose of a shock absorption. So when someone hits their face and facial bones, that having these spaces help the face to collapse, and that absorbs some of that force rather than that force traveling further. And the good comparison of that is the front part of the car, how it collapses in an accident. And it's designed to collapse because that act of it collapsing, that actually takes off some of that forces that are present in these sorts of run-ins. So that's speculated to be one of the functions of paranasal sinuses. Up to you whether you believe in it or not, but I do want to throw it out there as something to consider. And finally, paranasal sinuses, we're finding more and more that they have a role in immune system. We don't fully understand this, so this is something that we're still looking. But those are the four functions, four roles of the paranasal sinuses. What I wanted to say about paranasal sinuses as well, and this is not in your exam, but if you think we have these empty spaces, and I think I've mentioned to you a few times that I used to work, I've had many different kinds of jobs, and one of the jobs that I had was working with gender reassignment clinic, and a lot of, if there's transitions one way or another, and especially if it's from male to female, there's a sanding down bones and facial features. But because we have these paranasal sinuses, we can only sand down or thin these features that are very masculine on someone due to the bony structures only so much, because we can't puncture that bone structure and then have the sinuses exposed. That also makes these individuals much more vulnerable if there's an impact to their face. I don't know if that's really something to know about. That's not in an exam, but something to think about. I always try to connect as many things as I can think of what I've done. I recommend that you do the same. The final thing that I do want to mention about paranoidal sinuses, you remember, if you have already taken medical terminology, you would have seen this, and if you haven't yet, you will definitely get to see it later, that are, whenever we have a term, itis, it told us that there's an inflammation of some kind going. So if we have sinusitis, it's the inflammation of sinuses that's going on. And we had some pretty striking pictures, I think, in our chapter material showing how these sinuses can get filled with all sorts of material. They do really show up. And I think that that's all I can think of about paranasal sinuses, what to say. We did also find this little opening at the inferior and posterior part of the nasal cavity, which was the opening of the auditory meatus. Auditory meatus was the connection from the nasal cavity to our inner ear. So there's a tube that goes there. And one of the functions of these tubes is to balance the pressure differences. So you know when you drive up on a car or take a journey on a plane, your ears might pop and all that. If you swallow, if you open your mouth, if you do anything that allows the air to go there, that might help you to pop the ear back. So that's the function why you get chewing gum or hard candy to suck on or things like that. If you're changing altitudes, that might cause your ears to. Well, what else could we find? This next one is really, really a good one, an important one, and I think that we really should be familiar about that. So the functions of the nasal. Why do we have a nasal cavity? There's 3 important functions, and these all relate to what we want to make sure that we do to this air that's coming in from the outside world before it travels further down and all the way to the lung and to the alveoli of the lung. So we want to make sure that that air is filtered, and we already talked about that in terms of the hair at the nostrils, but also in terms of that mucous membrane lining that line the nasal cavity. That's great for catching anything that shouldn't be there, any particles. So the filtering of the air was first of the important 3 things that nasal cavity did. The second important thing is going to be warming up the air. So we remember this was a thin layer, not as thick as the skin, for example, and it was also rich in blood supply. You remember the rich nosebleeds and so on. And because there's a lot of blood supply, blood brings heat with it, and it's in a close contact with this air. So it helps to heat it up before it travels down further to our respiratory tract. And the final thing that we saw, again, we remember the mucus was kind of a humid, sticky, wet liquid. So that allows us to moisten the air. So you remember, again, we want to have that air to touch as much or spend as much time as we can in the nasal cavity and get as close to the mucous membrane there. That all adds up, that we get that air moist. And how you know that we have all of these things going on? Well, first of all, if you breathe in something and it doesn't get filtered, say you breathe in through your mouth, big gas of dusty air, you start coughing. Our nasal cavity didn't have a chance to do its job. That's why we breathe normally through nose, and we get a chance to filter this air. If you take a big breath through your mouth and breathe in a lot of dust, that's not ideal. You start coughing. The other 2 things that we notice, if you take a big breath, especially cold and dry air, say you go out on a winter day, you start coughing. That's because we didn't get a chance to heat up that air. We didn't get a chance to moisten that air. And all of a sudden we have this air that our body isn't designed, our respiratory system isn't designed for the lower parts of the respiratory system. So really those 3 functions I would know, and I would make sure that you can pick them out from wrong statements, for example. Well, the next part that I want to talk about with you guys is our pharynx. So pharynx is actually a skeletal muscle tube. It is located at the back and back of our nasal and oral cavity. So I'm looking, why did I write from the back of the mouth? I would say from the back of the nasal cavity. And it runs all the way to the beginning of the larynx, around at the level of cervical vertebra 6. So it runs quite far down. And we had 3 different parts for the pharynx. I might need to fix this part back of the mouth. I'm probably going to change it from the back of the nasal cavity. So in case you want to make an alteration to there, I don't really have a tool to write on this. So just so you know, I probably wouldn't use this term here. So I might change that for the updated version of the study guide from the back of the nasal cavity. Pharynx can be divided in 3 parts. So we have nasopharynx behind the nasal cavity, oropharynx behind the oral cavity, and laryngopharynx, which is the part that is right above the larynx. So it's the area, if you think of our epiglottis and how if it's upright, the space that takes, well, that space would be our laryngopharynx. I'm always, as an anatomist, trying to define where something starts, where something ends. If you're not into anatomy, you can focus on the physiological aspects. But I would know those three parts and kind of their order where you would look for. That would make a good exam. Let's have a look at our larynx. So I have a lot listed here. We're going to be focusing on the cartilages, but really larynx is made of other things than just cartilage. So like I've highlighted here at the very end, we'll go back if you're worried, just so you know. We do have muscles, we have elastic tissues. So cartilages aren't all that our larynx is made of, but it's what are the most anatomically striking structures. So we're gonna focus on those. And let's start with the first cartilage, which is the most prominent. It's the largest cartilage of the larynx. That's our thyroid cartilage. Where would you find thyroid cartilage? Where your thyroid gland is, so right behind that. And that forms this big structure that we often see. And one part of this big structure is our Adam's apple, the protrusion here. Often more prominent in men, but doesn't mean that it could be prominent in female as well. And the correct anatomical term for that would be the laryngeal prominence. So that's something that we can see or at least feel as part of our thyroid. And if you continue down your thyroid cartilage after you found your Adam's apple, you'll notice there's a little gap at the bottom of that. And then there's another hard structure. And this is kind of a ring that's actually fully closed ring. It closes all the way to the back of the larynx. And that's our cricoid cartilage. So it's usually easy to find your Adam's apple, follow it down the line of the thyroid, find the gap between thyroid and cricoid cartilage, and then find your cricoid cartilage. If you have totally forgotten what I'm talking about, I really encourage you to look at those diagrams that I drew or diagrams that are in your study materials in your textbooks wherever. So that's a fully closed, most inferior part of the larynx. Well, what else do we find in larynx? The next one that I've listed, and these are not in specific order, they're probably in just the order of my preference, what I think is fun. We have epiglottis. And epiglottis is a fun word, and it's a fun structure. It was this flap-like structure located inside our thyroid cartilage. And you remember the function of this flap-like structure was really to seal off our trachea once we have food or liquid that we're swallowing. So remember, our esophagus is going to be more anterior, and then our trachea is going to be more posterior. We want to make sure that no liquids, no food goes into our lungs, goes down to trachea. So epiglottis did the job for that. But remember that most of the time, epiglottis is open. And that again goes back to what we saw at the very beginning. What were the important things that we wanted to make sure? We wanted to make sure that our airway was open so that there can be breathing happening. So epiglottis is usually open or up so that this airway is open at all times. We want to secure that. And it only closes when there's a swallowing of food or liquid taking place. Well, what else can we see? And you'll also notice if there's a number and it's a single number, it means there's only one of these cartilages. But some of these cartilages, we have multiple. So let's have a look of the first cartilage where there's two of those. And these are going to be our arytenoid cartilages. So these are, again, inside the thyroid cartilage, inside that large structure here, a kind of a pyramid or triangular shaped cartilages. And the reason why I wanted to do these first ones is that these are moved by muscles, and this muscle movement of arytenoids helps us to tighten and loosen our vocal cords. So our vocal cords are attached to arytenoids. So it is changing the arytenoids location or angle that were able to manipulate the vocal cords. And we'll talk about vocal cords a little bit more in a sec. So 2 of those. And right on top of those, we have tiny tips known as corniculates. So tiny cartilage parts on top of the arytenoids. Again, I encourage you to look at the diagrams if you're kind of like, where is this? What are we talking? And the final structures, again, 2-paired structure that I want to mention is our cuneiform cartilages. You often don't really see these, and I'm kind of thinking, is that word within thyroid cartilage the best term to describe it? They're actually within the mucous membrane lining, within the tissue, soft tissue of the of our larynx. So cuneiforms, unless you're able to dissect it or imbue it, image it somehow, if you're just looking at the tissue without altering anything, it's hidden inside. Well, what's the function of our larynx? Let's talk about that a little. So there's a lot going on. One of the big ones that we want to remember, larynx is where the air flows through. It also provides, thanks to our epiglottis, the separation for the point for the air and food to go down our esophagus, our air to go down our trachea. Of course, as we already mentioned, the vocal cords, voice production takes place here. And if we think of vocal cords, what are they, what they look like, what does that tell about them? They're white in color. They're elastic fiber tissue. And the reason why they're white is that there's no blood vessels, there's no blood supply to the vocal cords direct. The space between, because we have 2 vocal cords, the space between them is known as glottis. If you want to use anatomically totally perfect language, you probably would call it glottis. And we had 2 pairs of vocal cords. Remember, more superior set of vocal cords or vestibular folds. These are false vocal cords. They're not really involved with the voice production, voice manipulation. The inferior, the lower set of vocal cords are what are our true vocal cords, so our vocal ligaments. So this is where actually the altering of the airstream takes place. Of course, remember that the voice production then happens in many other places above the vocal cords, so we still alter the voice. That's very important to notice, that it's not just the vocal cords that give us our voice. There's a lot of other things happening superior to that. But I think we're ready to jump on to have a look of trachea. You remember trachea was made of these C-shaped rings that were then connected. Trachea ran from the bottom of the larynx all the way to the mediastinum, to the carina, which was that point where we had our primary bronchi dividing, our primary bronchi starting, our trachea dividing into two primary bronchi. What we notice when we look at the primary bronchi, we notice that the right one of them is steeper. And this is why I think we often see this, especially with young kids, young babies or children, that they try to put everything they can find to their mouth. Sometimes they end up breathing it in. And if there's a foreign object, it's usually in the right lung because of the steeper angle of the right primary bronchi. Kind of jumping back, I'm kind of trying to catch up. What didn't I mention? You remember I mentioned these C-shaped rings that make the trachea. They're C-shaped because the posterior part of these rings is where the opening is. And this then allows us to make sure if we need to have, for example, space for the esophagus to extend. So there's a big food bolus that we're swallowing. So that gives a little space for the esophagus to kind of push in where the trachea would normally be. Clinical relevance, this is, I guess, every medical student's dream to get a chance to do tracheotomy. So incision to relieve if we have an obstruction higher up here. So we get the air supply down the trachea and down to the lungs. Usually you would look for this space probably between our between our thyroid cartilage and cricoid cartilage. That would be probably a good space. The thing that why we want to be careful about this, and why you actually need to know what you're doing, you need to be careful about the big blood vessels that run here, and you need to be careful about nerves. Of course, you also don't want to go too far. In an ideal world, only in hospital, only with sterile tools and people who have a chance to really assess that and so on. But that's not always the case in medical situation. So sometimes you see creative solutions being done. But tracheotomy was the clinical relevance that I could think of there. Then we continue from our primary bronchi to secondary bronchi, segmental, tertiary bronchi, bronchioles, terminal bronchioles, and we get to the alveolar ducts and alveolar sacs eventually. What we'll notice in this tract as we look at it, as we move from more superior parts to more inferior parts, and also from wider parts to more narrower parts, the amount of cartilage decreases, and the amount of smooth muscle increases. We also, what I do want to mention here is that remember the spot where the primary bronchi divided, that was our carina, or our trachea divided into two primary bronchi, but the spot where these primary bronchi actually enter into the lungs, that's hilum. That's a term to be aware of. And we'll come back to the term hilum. You'll see that also in a diagram. What can we see about lungs? Well, here I have some important terms that I'd like you to know about the lungs. And I have drawn arrows to point them out. I've also left a little bit of space underneath in case you want to make notes. So let's have a look what we can find. And we're going to start with apex. You remember apex of the heart was this sharp end, and that was pointing inferiorly and slightly to the left and anteriorly. Well, apex can be wherever located. Apex just refers to this sharp tip, sharp end. Well, in case of the lungs, apex is going to be the most superior part, so most superiorly. So it's going to be here. And it usually goes much higher than students or anyone, really, medical professionals, lay people think, almost at the level of the collarbone to the back. What else can we see? The base, we've talked about the base of the heart, but we also know this base of the lungs. Well, base is this inferior surface that rests right on diaphragm. And you remember diaphragm was our main respiratory muscle. So base of the lung is down here. It rests on the diaphragm. Hilum, that was the word that I mentioned that we'll talk about more. So hilum was this point where the primary bronchi entered into the lung. So those would be ones to know. What else? I have the root there, the vascular or bronchial attachment to the mediastinum. So, of course, lungs are attached to this middle space. Costal surface, well, that's where the ribs are, all the parts where lungs attach to their rims, these surfaces. You find it anteriorly, laterally, also posteriorly around the lungs. So those are some of the terms, some of the landmarks to be aware of. What else do we find? Quickly, this is kind of a revision, but I like you to be able to notice them if you're not familiar or have forgotten. Right and left lung differ. Left lung is much smaller, and that's because the heart is located more on the left side of the mediastinum. So heart takes a little bit of this space of where the left lung would be located. And we actually end up noticing this structure called cardiac notch. So a little bit of space is taken off the left lung because of that. And we can see the heart and some of the important structures of the heart as depressions on the left lung surface. Well, this results, because there's a size difference, in differences also in the number of lobes, in the number of fissures that we might see. Let's start with the lobes. So on the right lung, we see our superior, our middle, and our inferior lobes. Whereas on the left side, we only see the superior and inferior, so there's not middle lobe. The other thing that we'll notice that then these fissures, you remember fissure was depression of some kind separating parts. We have two fissures separating these three lobes, so we have our horizontal and oblique fissure in the right lung. On the left, only the oblique fissure. Notice how oblique kind of goes in this angle, whereas the horizontal really is almost straight in terms of if you think where the horizon is. This is something that I teach you and you learn, and that's on average the case. However, in reality, we do see variation in the number of lobes, in the number of fissures from one individual to another. Normally, when I do labs where we dissect catheters, there's usually maybe 5 catheters, sometimes 10 on display at one time, students work in groups on those. Out of those 5, out of those 10s, at least one would have a different number of lobes, a different number of fissures. So this is just what you get taught. This is the textbook, but in reality, there is variation. It's never quite as definite as our textbook and typical human body anatomy or physiology looks like. So remember that. But this is what we're going to learn, and this is how we assume that the lungs are made. We noticed that lungs were located within pleural cavity. And pleural cavity was made of 2 layers and a liquid space in between. Well, the innermost layer was our visceral layer, and the outermost layer was our parietal layer. Well, what's the function of this fluid and this space in between these two? Well, the fluid allows these layers to slide easily past each other. It provides a little bit of cushion, so allows all this movement for the lungs in this space that's otherwise fairly held tightly. So it would be hard otherwise for structure to move there. But one of the big things that we notice that this space also does, there's a negative pressure inside this space. And this negative pressure is really important because it causes lungs to be expanded at all times. Sometimes we might have patients who have had their pleural cavity punctured, and as a result, their lungs will collapse. So that would be a clinical relevance. You don't want the lungs to collapse, then they're not able to function, they're not able to do their full job. So do notice that negative pressure, there was quite a few slides. I think I actually gave you the values, how we could look at the differences. I think it was in partial pressures that we saw this negative pressure within the pleural. The big message there, keeps the lungs expanded. And then we get to our alveoli. So these little sacs, tiny sacs, and they're often as clusters of sacs at the very smallest, very end part of the respiratory tree. There's loads of those, about 300 million in a lung. And these are the site where the gas exchange happens. So their wall is very, very thin, and it often runs right next to, or capillaries run right next to these. So the purpose is that we make the exchange of materials between the insides of the alveoli and the capillary blood as easy as possible. So we keep that distance short so that by this partial pressure difference, by the process of diffusion passively, the exchange of the gases can take place. So I think that that's all from the anatomy of the lungs. Let's have a look of the anatomy of the respiratory system. Let's have a look of the respiration in general. And this is a nice diagram. I like it but it really is up to you what you feel that works for you and helps you. I know that I did draw it for you. So if you think that it helps you to follow with that, you'll find a larger one in your notes, but you can understand this even without this diagram. I just like to learn visually myself. So it's up to you, whatever works for you. But I want to go through phases of respiration, and there's 4 phases. Remember though that it doesn't mean that we only go 1, 2, 3, 4. We also go backwards from 4, 3, 2, 1. So air doesn't just flow into the lungs, it also flows out and so on. But let's have a look at these 4 phases of respiration. So we're really getting to the physiology. This is a little bit more artificial in my mind, but it's an important concept to be aware of. So the first part that we notice is our pulmonary ventilation. So this simply refers to what you see here, transporting the air from outside the body all the way to the lungs and all the way to the alveolar sacs of the lungs. This is a little small picture, but if you find the original reference or zoom in your notes, you'll notice. So pulmonary ventilation refers to this process of transporting air from outside the body to the lungs and to the alveoli of the lungs. We have 2 different kinds of breathing movements that we can do. Well, you can breathe in, inhalation or inspiration, whichever term you prefer to use. Think of, do you have to spend energy to breathe in or breathe out? Well, actually breathing in is the active process. So you have to recruit muscles. You have, for example, your external intercostal muscles, but the main muscle of respiration is our diaphragm. That's the main respiratory muscle. So what diaphragm did, it was this thin sheet of muscle separating our thoracic and abdominal cavity. And it's usually a little bit like this dome-shaped structure. Remember, at the very base of the lungs. And what happens when diaphragm contracts? It moves down and flattens down, which causes our lungs to follow and grow in size. At the same time, the external intercostal muscles also help our ribs to move a little bit forward and upwards. And all of this makes our lungs bigger, the space inside the lungs bigger. What did we learn about diffusion? The flow of the materials was always from high pressure to low pressure. Well, if we make this space bigger, that means that that space ends up having low pressure inside it because that same amount of air is now spread out in a larger space. So outside of your body is now going to have a higher pressure, or higher amount of air, so air flows inside to the lungs. So that's how we get air into the lungs. Remember, active process, diaphragm, main respiratory muscle. Then if you relax once you've taken a deep breath, automatically air goes out of your lungs. It is a passive process, mainly due to the elastic recoil of the cartilages, ribs return back to their normal space, and also the surface tension of the alveoli of the lungs. We'll notice that the negative pressure, again, ensures that the lungs are maximally expanded, but they automatically still end up being smaller because of just we've spent the energy to stretch the lungs bigger. When you relax, it returns back to its normal. Again, in this case, the size of the lungs decreases. Now, all of a sudden, this air that we had inside the lungs gets packed into a smaller volume, and that means that the outside is now going to have a lower pressure. Inside is going to have higher pressure because we're packing the same amount of air into smaller volume. That means substances always move from high pressure to low pressure. Air flows out from the lungs. I hope that helps to explain it. And finally, those two are not the only kind of ventilation movements that we can have. We can also have something called forced expiration. That's where we actually spend some energy. So it's an active process. where we really try to squeeze in our lungs and our chest cavity even smaller than what it normally would be. So we're recruiting new muscles. We're recruiting oblique abdominal muscles, transverse abdominal muscles, internal intercostal muscles to really shrink the lungs so we can force all the air out from there. So that was our pulmonary ventilation. Then we get to the external gas exchange. And as you see that we have two gas exchange sites. So this is the first. And this is where the air gets changed, or gases get changed from alveoli to capillaries, and also from capillaries to alveoli. So from the lungs to the cardiovascular system. And this is driven by this partial pressure difference. You remember again the same concept from high pressure to low pressure. So if you have more pressure in blood than you have in alveoli, substances are going to move from the blood to the alveoli. If you have higher pressure in alveoli than in the blood, then it's going to move from the alveoli to the blood. So partial pressure difference was the driving force for the gas exchange. And I gave you a lot of numbers and things to look at, but really what I want you to know is just the driving force. This is a passive process. This happens due to this gradient difference. And this is really, if you think, this takes place within the pulmonary circulation. So we're taking the blood to the lungs. So kind of combining what we've already learned before. Let's throw in some terms of that. Well, now we have oxygen that has been picked up from the lungs to the blood, and also we have gotten rid of the carbon dioxide in the blood to the lungs. Well, now we have to transport this blood somehow. So this is the gas transport stage. This is the systemic circulation, as is the next step as well, if you wish. Now we've gotten the gases to the tissues in the body. It's time to do internal gas exchange. So again, remember the directions where they go. This is because of the partial pressure differences from high concentration to low concentration. So oxygen leaves the blood, goes to the tissues, and carbon dioxide leaves the tissues, goes to the blood. It can be something else than tissues. It can be, for example, our interstitial fluid, not just the cells, not just the tissues. It can be other things, but by large, that's a good overview. And then, of course, everything moves the other way around when we get rid of carbon dioxide. So those are the 4 stages of the respiration, or 4 phases of the respiration. What else could we talk about? I mentioned here a couple or some non-respiratory air movements. So, of course, air moves also for other purposes than just breathing. Of course, our speech production, coughing, sneezing, crying, laughing, hiccups, yawning. There's a lot why we might move air. I don't know, maybe the yawning would go into a respiratory air movement. So I kind of might consider, should that be in brackets? I might change it to the next version, which is for the evening and on Wednesday's review session. So you might see this study guide being changed, but it doesn't really change drastically. You can just put brackets around the yawn. And we already talked about that. And I think yawning is a good one to know for next. So a couple of terms that I still want to talk in terms of the physiology of the respiratory system. Surfactant was this lipid protein complex that helped to reduce the surface tension and prevented the alveoli from collapsing. Whereas the compliance, I often think of compliance as the measure of stretch that the lungs have. So it really measures the change in lung volume with given change in transpulmonary pressure. I think the easier way is to think about it as a stretch of that the lungs have. Surfactant was this mixture of lipids and proteins preventing alveoli collapsing. Reduces also the surface tension. So I think that those 2 should be fairly nice ones to know. Let's talk a little bit about the ventilation and movements of the air as we measure it. And the technique would be, of course, spirometry. Spirometer would be the equipment that we use to measure how much air, the volume of the air, where is it moving? Is it coming in? Is it going out? And let's have a look what we can learn about the ventilation. So the first part, abbreviated as TV, was our tidal volume. It shows us this amount of air moved in and out of the lung with normal breathing. And as you'll notice, it's not a lot in the grand scale of what it can be. So in a normal breathing, we don't actually end up recruiting all of our lungs. We breathe some out, we breathe some in. This is known as our tidal volume, the normal breathing. You can breathe more in, so you can get more air in. This would be our inspiratory reserve volume. So you can actually get the maximum amount of air that can be inspired forcefully, so recruiting extra beyond the tidal volume. That's our inspiratory reserve volume. And you'll notice that it's beyond the tidal volume. So tidal volume is not included into this inspiratory reserve volume. Something to note there. That's why I changed my drawing a little bit to highlight that this is the extra that we can do if you wish. So abbreviated as IRV, inspiratory reserve. Let's have a look of the opposite, ERV, expiratory reserve volume. So this is the amount of air that we can forcefully expel. So this is this extra that we could do. We saw here some time ago. We saw it somewhere. Forced expiration. So as forced expiration here. So that would be what we get in the expiratory reserve volume. The extra amount of air that can be forcefully with forward expelled from the lungs. And finally, if we look at this last bit of air that always remains in the lungs, that never leaves the lungs, even though we might try to breathe out as much as we have some air left into the lungs. This is known as a residual volume. So the air that always remains in the lungs is known as RV, residual volume. Well, why do we measure these volumes? By measuring these volumes, we can calculate respiratory capacities. So let's have a look at those. And I have the same diagram that you saw before, but I have more stuff added to that. And we're going to start with inspiratory capacity. So that's all that you can breathe in, what you breathe normally in your tidal volume, plus the inspiratory reserve volume. So if you add those together, that gives you the how much you can breathe in, the value for it. What else do we find? We find a value for FRC, which is our functional residual capacity. Notice I have a R there, whereas I'm supposed to have F. Clearly, I hit the wrong button there. So I'm going to change that on my notes. So functional residual capacity was when we added our reserve volume, so all the air that never leaves, and then the maximal expiration together. That's our functional residual capacity. Let's have a look at the concept of vital capacity. Vital capacity is what we get when we combine our normal tidal volume, our inspiratory reserve volume, and our expiratory reserve volume. So that's all the air that can move at the maximum in and maximum out, plus the normal breathing. That's our vital capacity. And the total lung capacity includes all of these plus the residual volume, the air that never leaves the lungs. That's the total lung capacity. So it's a good thing to know how we get these volumes and how we get these capacities. So something to bear in mind there. And these are the diagrams that I mentioned earlier on, giving us the values for the partial pressures of different gases, oxygen and carbon dioxide, that were the driving forces for gas exchange. We'll have a look at that in a bit, but I do want to mention the concept of dead space. So you have two kinds of dead space. You have anatomical dead space, so that's all the passageways within the respiratory system where the air remains, regardless whether you're breathing out or in. That's just where the air is. We don't get all the air out. Alveolar dead space is the space occupied by the alveoli that don't function anymore. They're somehow damaged, they're collapsed, they might have particles there that shouldn't be, something's gone wrong with their surface, many different reasons. But if you combine the anatomical and alveolar dead space, that gives you the total dead space. So the amount of space or volume of air that really doesn't do anything, that doesn't serve a purpose in the lungs. And finally, I do want to talk a little bit about gas exchange. So this was what these diagrams that I included here were about. And they don't have colors, I'm afraid. So you can color if you wish. The color here for the blood would be bluish. It would be bright red here. And then blue again here at the bottom. So, gas exchange, we noticed that we had external respiration and internal respiration. As we reviewed here when we were looking at the phases of respiration, we had external gas exchange, internal gas exchange. So let's have a look at that a little bit. External respiration between the lungs and the blood, so between the alveoli and the capillaries around them. Internal respiration between the capillaries around the tissues and the tissues, or capillaries around interstitial fluid and interstitial fluid. So that's something to know. It was the partial pressure differences, as we saw, always going from high to low. We can look at, this is our alveoli, partial pressure of oxygen is 100 milligrams of mercury, I would imagine. And the blood coming from the systemic circulation had a partial pressure of 40. So which way is the oxygen going to move? It's going to move from high pressure to low pressure. So it's going to move from alveoli to the pulmonary capillary. The opposite applies for the carbon dioxide concentration. 45 milligrams of mercury carbon dioxide pressure in the blood from systemic circuit, in the alveoli, in the air around us, only 40. So carbon dioxide is going to move out from the capillaries to the alveoli. And you'll notice that the blood leaving this pulmonary circulation has the same numbers as we would have in the air in the alveoli. The opposite happens then within the tissues, within the interstitial fluid. You'll notice from 100, which was the oxygen concentration in the blood, to 40 in the tissues, so oxygen travels from blood to the tissues. Opposite applies, higher partial pressure of carbon dioxide within the tissues, within the interstitial fluid, whereas lower in the blood, so it flows from the tissues to the blood. And that takes us to the very beginning of our diagram that we saw. Let's have a look quickly how these different gases are transported in the blood. Most of the oxygen transported bound to the hemoglobin. So that's something to note. Only tiny amount bound to plasma. So hemoglobin is really the major oxygen transporter. Carbon dioxide, we saw the bicarbonates. Earlier on, we discussed about that. About 3/4 pound of carbon dioxide bound to the as bicarbonate. Then some as a hemoglobin, bound to the hemoglobin, some to the plasma. Well, what's the clinical relevance of all these if we think of these concentrations and exchanges of gases Well, the clinical relevance that I thought about here that would be nice to think of is hyperventilation. If you have deep, fast breaths, you might end up breathing out, removing too much carbon dioxide from your body. Body needs to have, or you might end up removing too much carbon dioxide. You need to have some. So what we do, we often give a patient in first aid setting. Not so much these days. This is kind of debatable because we're worried about suffocation. In the past, we gave a paper bag where they were breathing. So they actually trapped some of the carbon dioxide that they would breathe out otherwise, and they would get it back in their breath. So hyperventilation, a condition that relates to that. And that, I think, completes our respiratory system. So you'll notice that the main emphasis on this exam is going to be on respiratory system. We are going to cover the urinary system, so that's going to be important, but we're going to focus on the urinary system, mainly on the nephron, and then we have finally a little bit to talk about the fluid, electrolyte, and pH balance. If you have to go, I totally get it, but what I'm going to do for the sake of recording and sake of completedness, I would like to continue through our urinary system. If you have time, I'm very, very glad and grateful for that, but I totally also understand if you need to go. So let's have a look of the urinary system. And kidneys are, of course, the business end of all that. And if you look, there's a lot that kidneys do. One of the main things that we learned when we looked at the blood and cardiovascular system was that kidneys played a role in blood pressure regulation. In addition to that, of course, we learned when we were looking at the chapter on the fluids, they regulated the fluid and water volumes. And we noticed also in the next chapter that the solutes that are wherever the fluids went, the solutes followed, or wherever the solutes went, the fluids followed. So kidneys are responsible for all of these things. In addition, they're responsible, for example, for the acid-base balance, filtering blood, removing whatever shouldn't be there, red blood cell production regulation, vitamin D activation, many things. But those are really the super important ones to be aware of. If we look at the external anatomy of the kidney, and the reason why I'm probably moving this differently than what you see is that I have other windows open, so it's not all of the screen I'm able to see. So that's why I'm moving the screen in a partially different way than you probably see. But external anatomy of the kidney, they look kind of bean-shaped. That's the word that I often get when I ask students to describe them. Of course, bigger in size. And what we notice that they are on this medial side concaved. So here is where we find our renal hilum. So this is where ureters, also vessels, nerves, all these sort of things enter and exit. So blood vessels come in, blood vessels leave, nerves, so on. That's the medial side. On lateral side, you'll notice that the kidney is more convex, so larger curvature there. But we also notice that the kidney's external anatomy looks very smooth. That's because they're enclosed within a renal capsule. So that gives them this nice, smooth appearance. If you think of the terms that we've used before, we talked about peritoneum. So these are retroperitoneal. So they're behind the peritoneum in terms of location. And if we thought about it, we noticed that kidneys were way further back towards the back of your body than we often end up thinking. And that has other consequences. Again, I wouldn't ask in an exam, but for example, if you use electric blanket, you sleep it on, don't sleep on your back, you can actually overheat your kidneys. What else should we know? Well, we noticed that the left kidney, and again, remember, we always talk in relation to the patient's level, right? We noticed that left kidney is higher, whereas the right kidney is lower. Well, why is this case? Because on the right side we had the liver, so that caused the right kidney to be a little bit lower in terms of its location. Internal anatomy of the kidney, we end up finding the kidney has a cortex. Kidney has a medulla. So cortex was this superficial part, and medulla was this deeper part. Just as we've seen with other structures that we've studied earlier on. Within the medulla, within this deeper part, we found these medullary pyramids or renal pyramids. You'll see these kind of a triangular structures in the cross section that we can see here on this diagram that I drew for you. What else? We saw the renal pelvis. I think that we have talked about that a little bit. So that's the funnel-shaped tube that continues then as a ureter. We see it here. Flow of the substances through the kidney from renal pyramids to minor calculus, major calculus, renal pelvis, ureter. Really, we didn't spend too much time on that. And I think that the most important thing for me that you get out of the kidneys is really this next one, studying the structure and function of nephron. So this is really the business end. So it's the structural functional unit of the kidneys. And this is what all of the urinary system depends on. So we have a bunch of structures that we should know from a nephron. And we also have a bunch of movements of the fluids that we should know for a nephron. And I think one nice way to review this is if you have a diagram of a nephron, you can draw it yourself or you can use the one that I've given to you. And can we find all of these on this diagram? If you can, you're in a pretty good shape. It's important that we know the structures, so anatomy, so that we can start talking about the physiology, what happens. And let's start with afferent arteriole. Afferent arteriole was this blood vessel bringing in the blood to one single nephron. And efferent was this blood vessel taking the blood away from this nephron. So remember, afferent for arriving, efferent for exiting. That's my mnemonic. You might have a better one. But that's how I think of it. And arterioles were simply tiny arteries. So notice, although I had in my handout or in my lecture material color difference between here, these are both arteries or arterioles. So there's no really color difference in the blood. I just did it for the sake of clarity in the diagram. What else do we find? We find glomerulus. Glomerulus was this hard capillary network, capillary tuft, that was formed between the afferent and efferent arterioles, and that was surrounded by the first part of actual nephron, which was our Bowman's capsule or glomerular capsule. So if you want to make complete notes for yourself, you can write them down. I'm just not writing because I don't know how to find a pen on the PDF file. So that's the only reason in reality why I'm not writing them on this diagram. I'm just highlighting them. So you see sometimes different words being used, but it's the Bowman's capsule or glomerular capsule that encloses our glomerulus inside. Then the next part that leaves the glomerular capsule is our proximal convoluted tubule. So it's this thicker part of nephron immediately after glomerulus. It wiggles down here. And after that, we get the nephron loop, sometimes also known as loop of Henle. This is much thinner in its appearance, and it loops down and back up again. And after that, we have then our distal convoluted tubule. So again, growing bigger, more distal part that we might want to know. And that continued all the way until we get to our collecting duct. and multiple nephrons contribute to a single collecting duct. That was a little wiggly line, sorry about it. But you'll notice that all through this distance, we have these blood vessels running alongside the nephron's structure, alongside the loop of Henle or nephron loop, and also alongside the proximal and distal convoluted tubule. But the nephron loop is really the structure that we want to build. So I think that we have reviewed all the parts of a nephron. What we want to do now, we want to review the movement of the fluids. And I was wondering, could I change the color of this somehow? But I don't know how to do it. So, or I can't remember at this point. I'm sure I can pull it up somewhere. So we're just going to go with this. Yeah, I don't know how to do that. Share. We'll just go with this color. Let's have a look at the movement of the fluids here. So the first time that we had fluids moving in a nephron was when the fluids were moving from our glomerulus to the glomerular capsule or Bowman's capsule. This movement of some of the stuff being picked up from the blood, some of the fluid being picked up from the blood, was known as filtration. So we're filtering the blood. So some of the fluids get filtered here. After that, the second time where we see it is the reabsorption. Now some of the fluids move away from the nephron. Ooh, I don't know what happened there. Away from the nephron and go back to the blood. This is reabsorption. So we're taking it back. So it moves from the nephron or loop of Henle, nephron loop, to the blood vessels. Well, a little bit later, we change our mind and we secrete some of the fluids from the blood vessel back to the nephron. So there's a movement in reverse direction. And finally, whatever is left at the end of the process is going to be our excretion. And excretion is what went from the nephron to our collecting duct. So you'll notice the ABCD on this version that I've used here as a review sheet. Again, shout out if I go too fast, but I think you should be good. So this all gives us a concept of glomerular filtration rate, often abbreviated as GRF. If you look at blood works, you might see this value being recorded when you prepare for an exam of some kind. You might have that measured in other ways. But that really tells us how well your kidneys are working. So it tells us how much blood passes through one singular glomeruli in 1 minute. So that really is an indication how well your kidneys are working. And sometimes if you go to medical imaging, for example, or if you give drugs, if you do certain things, we want to know that that patient's kidneys are working. Because if not, we can't give a certain contrast agent or we can't give certain drug. So GRF is a good indicator of that and quite commonly used. The other thing that I want to mention in regards of the nephron is a hormone that we know from the past, ADH, our antidiuretic hormone. Well, what antidiuretic hormone did, it made us pee less. So lesser amount of urine is being produced. Well, how this happens, we knew this all from before when we reviewed hormones, but how this happens is going to be explained now that we know what a nephron is. Well, the wall of distal convoluted tubule becomes very permeable when there's high amounts of antidiuretic hormone present. So all of this, the sudden, this wall allows liquids to leave the nephron and go back to the circulation. So if a lot of liquids are leaving the nephron, we end up producing less urine in the end. So that's how anti-diuretic hormone works. And that's where it works. So I hope that that clarifies. I think earlier on when we talked about hormones, it kind of was left just floating in the air, not a real explanation. So really, the nephron is the business end of the urinary system that I want you to know. But I do want to talk about the other parts just quickly. You'll notice I'm not dedicating as much time for these, so that gives you a clue they're not probably as vital to know in depth. Ureters connected the kidneys to the urinary bladder, about 25 centimeters in length. They are also retroperitoneal, so behind peritoneum, made of smooth muscle, so smooth muscle tube, which allows this muscle movement that we're able to move materials along ureters, even against gravity. Even when you're upside down, the urine travels from your kidneys to the urinary bladder. That's how we're able to have our urinary system to work, for example, in space. So it's thanks to this smooth muscle tube. A urinary bladder. What does urinary bladder do? Only thing that it really does, it's a temporary storage spot for urine. So urine gets stored within the urinary bladder. Again, behind the rear peritoneum, retroperitoneal. It's located at the pelvic floor. Remember, this bladder can stretch and shrink depending on the need. So very much of a changing structure. Not going to be in your exam, but as a side note, you should not, if you have a patient who has had an impact to their especially abdominal area, if you suspect that there might be injury there, damage there, bleeding, things like that, you should not encourage them to urinate. Because if they urinate and empty their urinary bladder, that might generate extra space that then causes the injury to be able to fully exhibit itself. So we'll try to have them holding urine until we get them to medical. But we'll notice at the base of urinary bladder, so at the very bottom, the ureters don't really enter urinary bladder from the top. They go all the way to the bottom. So they go around and then enter to the bottom of the urinary bladder. So two ureters, so we have two openings there. Those are more posteriorly, and more anteriorly we have one opening. This is for our urethra, and we'll talk about that next. But these 3 points form a structure known as trigone. So remember, posteriorly the ureters and anteriorly the urethra. This structure might be something that you use as an anatomical landmark, for example, for imaging if you end up doing, for example, ultrasound or something. Finally, the urethra. That's the tube that carries the urine out from our urinary bladder to the external world. It's a muscular tube that has 2 sets of sphincters. Internally, these sphincters are involuntary. Externally, these are voluntary. So we're able to control our urination to a certain extent. However, if the pressure grows too high, then we might lose the control of the external sphincters. Also, age might affect, certain conditions might. But what we'll notice, urethra, the length of it varies from males to females. If you look at the male length, 17 to 19.5 centimeters in length of the urethra, you might think that that's kind of a long distance. Remember, some of it travels inside as well. Females, about 4 centimeters. Remember, urethra for males shared by the reproductive and urinary system, whereas for females, only urinary system. Females, because the length of the urethra is shorter, they end up developing more likely UTIs, so urinary tract infections, because of the simple fact that the distance for the bacteria to travel is shorter in females. So we see how the anatomy correlates with pathology. These things all we need to learn together. We can't just focus on anatomy. We can't just focus on physiology, and we can't just focus on pathology. They're all linked together. And finally, I think that I've just mentioned here just a fancy word for urination, micturition. That is a good one to know. Again, proper medical language, proper anatomy, proper physiology terminology. So we want to encourage you to use that. So rather than talking about urination or talking about peeing, we talk about nutrition. And I think final thing that I discussed was the composition, or I think I discussed this in a different order, but I added it here. The composition of the urine, major part of the urine, most of it water. Some solutes, nitrogenous wastes, urea, uric acid, creatine, and then I've got a bunch of others listed there. What we really want to know about urine is that if there's too high concentration of any of these, then we get concerned. Or if there's abnormal solutes that we don't expect to see. What would be abnormal if we have a lot of blood proteins, if we have white cells, if we have bile pigments? All of these would be signals that something's there that shouldn't be, and this might be an issue for us to be concerned, and they can signal different things. So this is all about the urinary system that I wanted to say. And the final chapter that we had a look was this fluid, electrolyte, and pH balance. There are going to be some questions about this, but it's not as heavy in emphasis as the respiratory system and the urinary system. So I'll mention that as well. If you are able to stay, I'm really glad. If you're not able to stay, I totally understand this review session is running longer than I expected. But I guess that it tells us that we're covering a lot. We're getting ready for the exam well. So there's going to be a recording if you have to go, but I'm glad if you can stay. So let's go through the fluid, electrolyte, and pH balance chapter. We notice that this is really understanding this is important so you can understand what is abnormal if we have issues with this. And we had a look of the water content. We noticed that water was vital. Water is vital in many ways for our physiology, but also in general life involved in water. All living things are mainly made of water. When we go to the space, we look for signs of water or presence of current water, for signs of life having been, or signs of current life. There cannot be life without water. So that's water is important. And let's have a look at how much of you is made of water. That varies depending on the source a little bit, between 60 to 75%, depending a little bit. Males tend to have a little bit more water in their body, females a little less. It would be good to know why. Well, females have a higher fat content and less skeletal muscle. And we notice that actually fat contains a little less water than skeletal muscle. So this would make a nice exam. There's the question for the written part, which is the extra credit question. But I'm not sure if it's going to be this one. But why are there differences between males and females? There's also differences between infants and elderly. If you look, infants have way more water in their body. Again, low body fat concentration. You remember, fat didn't contain a lot of water. Low bone mass. So again, that's something to bear in mind. Older people, then less fluids in their body. I think I had on this slide a comparison of a grape and a raisin. That's a little extreme. But if it helps you to remember, you can remember through that. Where is the water located in the body? Most of it is within the cells. So it's intracellular. About 2/3 of all the fluid. 1/3 outside the cells, so extracellular fluid. Extracellular fluid contains all the interstitial fluid, blood, plasma, lymph, all these things. But really, the most of the fluid is inside the cells, some outside. Let's have a look at the characteristics of water. This should be revision from BIO201 or BIO156, whichever one you took. But why is water special? Why is water so important for us in biology? Why is water essential for life? There's 7 characteristics that I've listed. We'll go through them quickly as they should be revision, but they are important ones to know. So water is a great transportation media. So it carries a lot of different stuff. Water can be used to transport a lot of different things. If you think of our blood, for example, most of the blood is water, and in the blood we were able to carry a lot of things, gases, nutrients, waste, hormones. Same goes for urine. Urine can carry waste products, a lot of things. So water is a great way of transporting stuff. Other thing that we notice that water has, and these are terms that students often know, but I just want to make sure that you know the right way around. Water has cohesion and adhesion. Cohesion refers to the attraction of water molecules to other water molecules. So water molecules are attracted to each other. So that's what cohesion means. Adhesion means that water molecules get attracted also to other surfaces. So when you look at, for example, of a glass that you overfill, the fact that it forms this dome that's higher than the surface of the outer highermost top point of the glass is because all this water at the very dome is sticking to other water molecules. So there's a lot of cohesion going. But also, if you look that you might have on your car's window, for example, water droplets, they don't really disappear from there. They're sticking to that water because water is great at attaching also to other substances, other materials. That's adhesion, an example of that. Well, we also often talk that water has a surface tension, high surface tension. For example, bugs can walk on water, on the surface of the water. Well, this is because at the surface of the water, we have high cohesion taking place. So these water molecules are tightly attached to each other. The weight of the bug cannot break this attachment. It forms almost this rubbery, elastic surface that sticks together tighter than what the force that the bug can put onto that spot. What else we noticed? Water is great in storing heat. So living on the coast, for example, you would see that the changes in temperatures would be lesser because the big body of water, whether it's a sea, whether it's a lake, it stores heat during the day, so it doesn't quite get as hot. And during the night, it lets heat go away, so it doesn't get quite as cold. So the reason why we get these extreme temperature changes in Arizona, from hot Arizona summer day, midday, to cold Arizona winter night, even during summer, is because there's so little water in the air. There's not that much humidity. So we don't have a lot of this heat reservoir taking place. Water can, of course, take different forms. I think that many students are familiar with the solid, ice, liquid, as we think of water in a glass, and of course, are gaseous water, so water vapor. There is a fourth form, plasma. We'll leave that for just a mention. Water is also great as a solvent. It's actually described as a universal solvent. So a lot of things dissolve into water. Maybe that would be better to put here than carrying solutes. A lot of things dissolve into water. And because a lot of things dissolve into water, and water is great at transporting these things that have dissolved in it, it is really essential for life. And it is really what we use to move stuff within the human body, within plants, within various other settings. Some materials don't dissolve to water fully, and we are not really interested on those on this chapter. We're interested on the ones that dissolve because when they dissolve to water, there is a change in charges a lot of time. You notice that they become slightly positive or slightly negative. And this slight difference in positivity or negativity is going to be important for us when we kind of look further these elements. They now become our electrolytes. So they carry a charge. And that affects a lot of other processes. So that really summarizes the key characteristics of water that we should be familiar. But if anything from this chapter, I would like you to learn this idea. And this might not be the most elegant way of phrasing it, but it's the best I could think of how to highlight the fact that, remember, if water travels from one body compartment to another, electrolytes will follow because of this, that these dissolved elements or molecules were easily transported by water. So if water moves from one compartment to another, usually we're going to see that electrolytes follow it as well. Or saying if electrolytes travel from one part to another, water will usually follow it as well. So this is why water and electrolytes are interlinked. They're linked to each other. They're connected. When we study physiological processes, one affects another. And we'll come back to this concept when we talk about tonicity in just a few moments. But what I want to say, I want to talk about the amount of water that we take in. It should be equal to the amount of water that we lose. So let's have a look at this concept. So in an average individual, we take in a day about 2.5 liters of water, and we lose the same amount. This is how homeostasis is maintained, same amount of both. Well, what is the biggest source of getting water into our body? Well, that's going to be drinking. About 2/3 of all the water, about this 2.5 liters that you take into your body, is through drinking. So the sense of feeling thirst is what's the main force that makes us get water into the body. We also notice that some water or some fluids we get from moist food, some as a byproduct of metabolism. You remember when we looked at these formulas, we noticed that water was produced as a byproduct. I might have to scroll too far and I might give up, but we noticed this when we had a lot of various chemical reactions that happened in the body. Usually water was produced as a byproduct. For example, in cellular respiration here, you notice we end up producing water. So some is produced also through these reactions, but that's a small amount. Mainly through drinking is how we get our water, through feeling thirsty. What else can we say about water? We can say how we lose water. Well, we lose water significantly by urination. So about 2/3 of all the water that we lose from the body is through urination. Some does get evaporated from the skin, also as a sweat. We breathe out some humidity, a little bit through feces. But really, thirst is the main way of getting water in, and urination is the main way of losing water, getting rid of water. And remember, these would be overall imbalance. Then we talked about this concept of osmoreceptor ADH mechanism. This is a concept that can be explained in very complex ways and can become very, very messy. I wanted to keep it simple, and I reminded us of this main concept that we've talked a number of times, homeostasis, and how homeostasis was all about maintaining consistent internal environment. The reason why remembering this is important, it gives us this diagram that we've seen before about homeostatic control mechanism loop. How there's some sort of a stimulus, some sort of a change that's sensed by a sensor, and that sensor passes a message to our control center, which was usually brain. And within there, we have a set point. What's the normal values? What we expect it to be? If this stimulus is off from our set point, we act on it through an effector. Some sort of a reaction happens. And usually that reaction aims to stop the stimulus from happening. That's negative feedback in homeostasis. That's a negative feedback loop. Only very occasionally can I think of examples of positive feedback mechanism. For example, when childbirth takes place, when lactation takes place, when clotting of the blood, forming of a blood clot scab takes place. So those are the only examples of positive feedback, where a stimulus triggers for more of that to happen. So most of the time it was this loop. And we went through this osmoreceptor ADH mechanism by using this loop. For the interest of time, I won't go through it step by step since we have this nice diagram that I provided for you here. I do encourage you to refer back to the lecture video if you feel a little confused. How do we make sure that if there's too much water in the body that we end up peeing it out? What are the steps that lead into that? If you are confused, what do we do to preserve water if there's too little water in the body? What causes us to pee less? Again, there's an explanation with this diagram, and you can refer to the lecture video if you feel confused. But that takes us now to a concept of tonicity. And the reason why I want to talk about tonicity is because it really ties in what we were saying here earlier on, that wherever the water goes, electrolytes follow, or wherever the electrolytes are, water will follow. These two are interlinked. So tonicity tells us about what happens when there's either a balance or lack of balance between where the electrolytes are and where the water is. So we have three cases that we'll have a look. We have our hypotonic cells, isotonic cells, and hypertonic cells. We're going to start with hypotonic. And hypotonic was when we had a cell that was from the inside, there was a lot of electrolytes. Outside, there's less electrolytes. Well, we remember wherever electrolytes went, water followed. So if we have this situation, water will start flowing into the cell, and this causes the cell to grow bigger. So cell can end up swelling. It can swell so much that it eventually even bursts. So hypotonicity referred to water flowing into the cell. How I think of this, and I think I told this in the lecture video, is whenever you can come up with a mnemonic, you remember it better. I think of hypotonicity. We have hypo sounds a little bit like a hippo. Hippos are these big, swollen animals. Well, hypotonic cells are hippo cells. They're big and swollen because water is going inside them. That might be a little naive, but it works for me. The second situation that we might have is that we have isotonic cell. In isotonic cell, the amount is the same inside the cell and outside the cell. So there's really no need for water to go in or go out. Some water will always go in, some water will always go out, but overall, the net gain or loss is zero. It remains the same size. It doesn't gain more water, it doesn't lose more water. How I remember this, when I think of isotonic, it makes me think of a sentence, I so perfect. If you're perfect, you don't need to change. This cell is telling you, I am so perfect. So isotonic cells are perfect. They don't change. They remain the same. And finally, our hypertonic cells. In these ones, there's more electrolytes outside the cell than there is inside. And as a result, the fluids flow out from the cell. So remember again, always where electrolytes are, water will follow. So water flows out from the cell. As a result, cell shrinks. It can even dry up at the extreme case. You think of again that grape that turned into a raisin. That would be an example of the fluids leaving the cell. How do I remember this? I think of hypertonicity. When someone's hyper, they have a lot of energy. What do you do if you have a lot of energy? You run around a lot. What do people who run around a lot look like? They're very skinny. So hypertonic cells are skinny cells. So hopefully those will help you to remember it. I don't know. They work for me. Really how this relates to what we've been talking about fluids is the recovery drinks or electrolyte drinks that you might see sometimes. So this is These are the drinks that we use to replace the electrolytes that you might have lost while you exercised, for example, for a long period of time and sweated a lot. You might have been drinking water, but you've lost a lot of salts. And if you don't replace those, you actually get really sick. And also, we need to have some electrolytes in your body so that you can take up this fluid. If you don't have any electrolytes, the fluid uptake doesn't happen. So it's really important that if you're exercising heavily, losing a lot of fluids, replacing them with water, you get some electrolytes as well. That's why, for example, if it's a hot summer day, you end up sweating a lot, you end up drinking a lot, you get a craving for salty stuff to replace the salts that you've lost. Well, this is the last thing that we're gonna look. I promise. We're gonna look at the pH balance. And that reminds us of the basic concept of the pH scale. You remember pH scale measured the concentration of hydrogen ions. This should be, again, revision from your BIO201 or BIO156. It's the hydrogen ion concentration that we're looking. Hydrogen scale ran from 0 to 14, 7 being neutral. And if we have pH that's higher than 7, we have a high concentration of hydrogen ions. That's alkaline or basic substance. If we have pH that's lesser than 7, we have low concentration of hydrogen ions, that's acid. Well, how does this all relate to us in our biology? We have normal ranges. For example, for pH, arterial blood pH is normally about 7.35 to 7.45. That's a very narrow range. That's because most of our body processes, most of our metabolic processes, most of enzymes, most things work ideally within this range. And our body maintains it with extremely great success. And if you look at the survival range, if we're anything less than 6.8 or if we're anything higher than 8 for pH, you're probably going to die. That shows that we still need to keep it within a very narrow range. And how our body is so successful in doing this is that we have 3 different methods that we can resort. Well, the first and the quickest way of dealing with changes in body pH is by using buffers. These are chemicals that end up binding to whether there's acids or bases coming into your body and trying to change your body pH. These chemicals bind into those and neutralize them so that your overall pH remains the same. And we use, at least in my BIO181, we do a lab where we experiment different kinds of chemicals. What would be good buffer? What would bind things really well? Acids and bases. So buffers were just chemicals that acted really fast, almost immediately, to prevent changes in the pH by binding into these acids or bases. The other way how we can deal with pH changes is through respiration. So funnily enough, we talked about a chapter of respiration. This takes about minutes to work, so anywhere from 1 to 3 minutes or so. And this is where we remove these chemicals that can make your body more acidic or basic through breathing them out, or breathing chemical components out, through breathing hydrogen ions, for example. The final way how we can deal with the changes in pH is through renal mechanism. So through your kidneys, by urinating out what shouldn't be there. We saw, for example, hydrogen ions were mainly urinated out from your body. This can take anywhere from hours to days to happen. So it's much slower than the 2 that we've saw before, but it's also most potent, it's most efficient. So, and strongest in terms of its effect. So these 3 processes allow us to make sure that we actually maintain our body pH so regular, so consistent. And we have clinical situations if we have too high or too low pH. That then comes with all sorts of consequences. And those are covered quite nicely in your lecture video. final concept I'm just going to mention it is our fluid therapy. So sometimes when you go to a hospital, that's quite common, we give you a drip. And that drip contains sterile fluids, but it also contains electrolytes in just perfect proportions so that your body electrolyte balance isn't shaken, or we can bring that electrolyte balance to normal. We do that normally to maintain your blood volume. If you have lost a lot of blood, for example, or a lot of fluids, for example, through bleeding, or you have had extreme diarrhea, so you have lost a lot of fluids through that way, or some other reason, fluid therapy, the drip brings that up. The other reason why we use fluid therapy, it's easy to add stuff to that if we want to bring it into the body. So again, goes back to the concept that we talked about, that water is an excellent transport media, and it also is great at dissolving stuff into it. So this shows up that why we might put the drip, well, because these fluids, water provides us so many benefits in achieving our goal, whatever we want to achieve with that drip. For example, administering you some substances through the contents of the drip that we administered. But it must contain the electrolytes. So this is the review guide. I know that it's a lot, but really it's only 3 chapters, and 2 of them are super important. The third one is important, but you've seen my focus is how I'm emphasizing things. I'm hoping that review sheet helped you. I'm going to stop sharing that and jump back to our PowerPoint. But of course, if at any point you didn't catch something or you're missing something, just let me know. And I'm also very, very happy to put it back on the screen. There's going to be also this video available for you later on. But we've reviewed the study guide for exam 3. I did at the very beginning more lab practical study guides. I think that there's some pretty good ones already up there. I might see if I get a chance to do even a little more. Remember, there's a practice option for the lab practical 3. Make sure you give it a go. You should do it multiple times so you see all of the possible questions, all of the possible pictures that show up. And I looked through the lab practical 3 already a couple of weeks ago, and there was just a lot of structures and a lot of more detailed parts of the structures, and not that much histology, a little bit, but it's really kind of remembering those detailed parts you can spot. I'm going to upload, now it's midday, later this afternoon, the lecture handouts, the lecture slides, not exact ones that I use, but very similar from your publisher to the Canvas course as well, if you feel that helps you with studying. Remember, I'm always very happy to reopen assignments. Let me know the details and we'll get you those points. There is tutoring available through the college, through the Cranium Cafe. I'm happy to also help. We'll take time for any questions in a second, but I think we've covered everything that we said to cover, but this is what I really want to remind you if you are joining now to the video or for some reason didn't see it before. Remember that the due dates have been adjusted a little bit. You have a little more time for doing last week's and the previous week's material. I really want to give you a chance to get through those. They are available until the end of this week. And the exam and lab that we are supposed to be doing this week, it's available this week, and it's also available next week. I want you to have a chance to finish the work first so you can take an exam on it. I don't want you to take an exam on something just cold without having had a chance to practice that. Hopefully that will help you. Really, I think that's all I can think of, but I'm so happy to answer to any questions that you might have. I'm happy to answer them while this is being recorded, and I'm also happy to stop the recording and answer any questions if you don't want them recorded. But in terms of my part, this is all that I can think of that would be vital to share with you So what I'm going to do now, I'm going to wrap up this recording here, and we can do if you have any questions. Let me stop.